So for this section of the chapter, we're going to look at genetic variation, and we're going to do it in sort of three different ways, and you'll also be able to practice this as well. So with the first part, I'm just going to verbally explain it. Hopefully it doesn't get too boring, but you'll want to go ahead and take notes. And as you take notes, I want you to think about how each one relates to the other. Because what we're going to do during class is this second part of it. And I want you to think about in your mind um, how these relate to each other, and you're going to create a concept map. And you can focus on or think about that right now because I'm going to be telling you like the definitions and have an image that goes along with it. And then when you come to class, you can kind of have some, stars or highlights of how you think they relate. And when you do your concept map, it's really important to recognize that not everybody sees the same connections, and that's okay. If you think about how they relate to each other, you will be more likely to remember them long term. So I do have a couple of different ways that you can practice these. So the first one is on the PowerPoint. I'm going to show you everything all at once here, but if you went through the PowerPoint, what you would see is that this would show up first, and then when you advance the slide, this would show up, and then the image would show up. So you can kind of practice that on your own. It's like pre-programmed flashcards for you if you want to do it that way. But so to get started, indels are kind of the easiest one to think about definition for. It's either an insertion or a deletion of a segment or DNA nucleotides. And these can have an effect on the protein because if you insert a section of it, you're going to have an extra amino acid, especially if you insert 3. If you insert 2, or in this one, sorry, if you delete 2, you're going to have a frame shift that's going to lead to a different coding in the protein. So you can insert or delete a small section, or you can insert or delete a larger section. And when we have a full gene or a set of genes that there's an extra copy or a missing copy of, we just call that copy number variation. So just in reference to the genes instead of smaller pieces of the genes like in the indels. That can happen because of differences in crossing over, duplication, like other types of duplication events. But if you look in this one, you can have unequal crossing over of these so that one of them gets an extra copy of the B, and that means the other is missing the copy of that B gene. So copy number variation, relatively simple. And gene family is just a term for a group of genes that have that common evolutionary origin. And in general, they often have similar functions or structures for their proteins. And basically, you had a duplication event that led to multiple genes that were originally coding to do the same thing. So a family of genes are very similar, just like you and your family might be a little bit similar, but have different overall functions. They don't have to do the exact same thing, but they originated from the same gene that got duplicated, and then over time, individual changes happened. Now, when we talk about, there are 3 different things that we're going to talk about in this kind of family, and I use the same image for each one. And just in case you're following along, you may have heard of gene duplication already. So one of the very common things across all of these is you have one gene, there was duplication, and that led to different functions. And so for this one, the paralog, and here's your mnemonic for remembering this, is that a pair of dogs have a paralog. So the idea between the paralog is that within the same species, you have a protein that does one function and a protein that does a different function, but they had this same origin. So in this one, within the mouse, the paralog is right here. You could also say that a pair of frogs has a paralog. In this one, it's a little bit more confusing because there's one over here and one over here. But this alpha, um, in this beta globin, this frog alpha and this frog beta would be a paralog. Now, a little bit different is that currently we've talked about genes that have a function. Pseudogenes are genes that originally had a function, but then something happened and it lost its function. So it's like pseudo kind of means fake, right? So it's a fake gene. It looks like it should be a gene, but it doesn't actually do anything. So it's a term, these non-functional copies of genes because of mutations that have happened and then they are useless now. And so there are many different possibilities that could lead to a pseudogene. And we'll learn about promoters later. This is before the gene actually starts this promotes the transcription of the gene. But if you have a missing or mutated promoter, if you don't have your start site, that's not going to do it. If there was a weird frame shift that happened, or if a mutation occurred that put in an early stop codon, or you're missing some of the introns, or the whole thing just got partially deleted. So a pseudogene is just something that used to have a function probably, but then got disrupted and now does nothing. Now, Hox genes are something that really do something. And there's a video on the evolution of Hox genes. And basically, the Hox genes are evolutionarily conserved, and they tell what proteins to get expressed where, so you have specific body specification. One of the interesting things is that if you take a fly and you put like these Hox genes, the UBX ones, but you express them over here, then you'll get a leg where an antenna should be. So it's just a series of genes that are responsible for expressing the proteins that are important for the specific body segments. Now, a homeodomain, this is a little bit, there's a couple in here that relate to this, but the homeodomain is a part of a protein that's going to bind into DNA. So hopefully you're able to recognize that this is DNA. So we have our double helix structure here, and we have the major groove within that DNA. And this protein, this little purple ribbon structure, is the structure of a protein. And this ribbon right here is able to bind into that pocket of the DNA. And so this part of the protein is called the homeodomain. I put a note down here that the homeo box is the portion of the DNA that coded for this, or the portion of the DNA that codes for the homeodomain part of the protein. So it kind of gets a little bit confusing when you're talking about, well, this is the DNA and this is the protein, but this protein was also coded by its own DNA. So homeodomain, or proteins with homeodomains, can bind into the DNA. Now, gene divergence, one of the things that is a little bit of a theme is that you have those duplicated genes. And then if you have two genes that are doing the same thing, if one gets mutated, then it will potentially have a different role in the future. So that is two genes that kind of started similar and then they diverged or changed in different ways to give them different functions. And you can see this, oh, that again, if you're looking for connections, um, that's what leads to the gene families having more functions within one gene family. And this is the best image that I could come up with. And it's basically just you have one original gene in all three of these. You don't have to memorize any of this, but I liked how it showed you different ways that you could achieve gene divergence. So you have or the same gene for at the beginning of all these, and then at the end you have a second gene because of hopefully what you see in the kind of, I'm not sure if it's like a darker blue or a purple, but these duplication events. And the duplication events can happen at different times, but you have that duplication event and one or both of the genes slightly modify to give you changes, which are represented by these little lines, these colored lines here. Those changes lead to changes in the gene, which lead to changes in the protein structure, which lead to changes in the protein function. So that is the gene divergence. You have the duplication, and then they diverge and change and give different functions. So similar to that homeodomain, this is a more general term, but protein domain structures. So the homeodomain was really specific, and that's the part of the protein that interacts with the DNA. These protein structure domains is just saying that there are multiple sections of a protein that can have different functions. So when we look at this blue section, hey, look, that's a DNA binding domain. So that would be a homeodomain. But then this one is going to break this. So this ATPase domain is going to affect ATP. And then I honestly, I don't remember what TPR stands for, but it does something. But and we just think about for a protein and a protein structure, it has a specific shape. Those different areas of that protein can have different functions, even though it's on the same protein. So all it is just a specific region of the protein that does something specific. And so these kind of tie together because you have the protein structure, and this is also just a different way to represent protein structure, and a change in the amino acid sequence. So again, in the class, we'll learn that, and hopefully you have this a little bit, but proteins are made up of amino acids. And those amino acids are coded by the nucleotides from DNA to RNA. And if there's a mutation in the DNA, they're going to lead to a difference in the amino acid chain. And so when we are looking at this particular protein, you can see that there's a difference for the duck, there's a difference for the turkey, there's a difference in the quail, and the pigeon. And so all these little different amino acid changes are giving a slightly different structure, slightly different function potentially for across different organisms and even within the same species. And just in case you were curious, because I was curious, well, where did I find this image? Because I found it a while ago. I was like, what is the difference? What is this protein, and what is the difference when we're talking about the different birds? So this is the MCR1 receptor. It's the melanocortin receptor, and the different mutations are what can give different skin color and different feather color for birds. And one of the things that we'll talk quite a bit about in this class in terms of, how the DNA leads to the expression of the protein. Um, you can have differences in the actual uh, amino acid sequence of the protein, and you can also change whether or not that protein gets made. So if we think about the dots on the last part, right, that's these individual amino acids, and there can be a change in the DNA. It's going to lead to a change in the specific amino acid sequence. But there are other things you can do to change the expression of the protein. So whether it's there or not. So you can change it at transcription, you can change it at the RNA processing, you can change how the RNA gets out of the nucleus, and then you can also change if whether or not the ribosomes are going to bind and lead to translation. So um, when we think about a variation in a specific sequence to change the structure of the protein, right? That's one way we can impact a phenotype of an individual. But we can also have all of these other regulatory steps in there as well. And another way that we can have differences is, well, many times when we talk about going from RNA to mRNA, we say, okay, so, and I'll quiz you on this real quick. Which ones get spliced out and which ones stay in? Right? The introns get spliced out, the exons stay in. And so that's kind of the really simple way and how most low level things explain it. But it's science is always harder than you actually think or honestly are originally taught, because we make it complicated with something that's called alternative splicing. And that's when, if you look at this one just has exon 1, 2, 3, 4, and 5. Done. And that's how you are originally told that that's how it works. However, in this one, if you look, now we have exon 1, 2, but we're missing exon 3. And then we have exon 5. And so we can have little things that are going to bind here, and we'll learn later that you can have other short sections of RNA that can prevent the RNA polymerase from reading this, and then, or recognizing that this is a transition point, and it will skip over it. So you get that exon 3 is spliced out. This one gives you the example where exon 4 is also spliced out, and I wish it would give you a different example where one of the exons actually gets spliced in. Or sorry, one of the introns actually gets spliced in. So you could have a longer, so both of these represent shorter where you've skipped an exon, but you technically could have this, and let's say that we put in intron 1 gets inserted in here. And so now you have, instead of shorter proteins, now you have a longer protein. So splicing variation or alternative splicing can give you many different possible proteins from the same DNA sequence. Retrotransposons. And so if you have stuff like that that can move anywhere, then every now and then it's gonna put something new, put something into place, and something new is gonna happen. So that's what these novel genes are likely coming from. So one of the things that we talked about in the previous chapter was parts of the DNA that are going to code for RNA that aren't necessarily going to code for a protein. So these are these non-coding DNA sections, but they do code for RNA. And so if there are changes in those sections, so these non-coding regions, if those are changed, then you can change regulation. And a couple of the things that this was the best thing I could find. It's just showing you that these little red bits are different types of RNA. and how they can affect either um, things binding to the DNA that's going to lead to transcription, or we're going to be able to stop the transcription, or those can bind in and affect the splicing at different sites. And then actually, these LNC RNA, so long non coding RNA, can go and do many different things. And we are recently starting to see that those can relate to disease as well. And later in the semester, I'll show you a specific example of that when we talk a little bit more about these. Right now, we're just highlighting kind of specific things that can affect and lead to differences or variation. So the long non-coding RNA that I just mentioned, that is just a specific non-coding thing it's just bigger. So it's just longer than 200 nucleotides. It has a secondary structure and it can go and do a whole bunch of different things. And specifically, those different things have to do with regulation of proteins. Now we're back to this image. So I mixed them up in here, but hopefully you do see this connection. So an ortholog is different than a paralog. So remember, a pair of dogs has a paralog. So the same different proteins within the same species that have a similar function or a similar origin. That's a paralog. But an ortholog now is something across multiple different species that have a similar function. So all of the alpha chains in frog, chick, and mouse have a similar evolutionary origin, and those are called orthologs. And so those will have similar functions within or across the different species. And now you have the third part, and that is a homolog. So homo means same, and the homolog part would go here, right? So homolog is just the umbrella term for orthologs and paralogs. And here's your cheat for how this particular image labels it. Okay, now we get to polyploidy, which is a wild thing. So generally, if you can pick the one that humans normally have, how many copies of each chromosome do we generally have? Right, and hopefully you're like, oh, well, we have two copies of each chromosome. But that is not the case. And plants can get really nutty where they have multiple, and I don't understand. Yeah, it's a challenging thing to think about how that actually works. But polyploidy, instead of like us, where we are diploid, and our sex cells are haploid, many plants and other organisms are polyploid, where they have multiple copies of the homologous pairs. So it's not just a homologous pair, but it's a homologous quartet in some. And gene duplication events, hopefully you see that this relates to a lot of the other stuff. So those gene duplication, we so that copy number variation is basically a type of gene duplication. And we also mentioned like the retrotransposons. So retrotransposons can lead to gene duplication in inefficient crossover can lead to more gene duplication as well. But these gene duplication are going to affect the size of the chromosomes. And if when you have the two genes, anyway, just see how this relates to a lot of the different things. I'll leave that up to you. And then I think that's it for this chapter. And like I mentioned, we'll practice this in class, and you will have the time to make your concept maps in class as well.